Activating molecular targets to reverse aging, a promising frontier in biomedical research. In the realm of biomedical science, the quest to reverse aging has long been a tantalizing pursuit. Over the decades, researchers have uncovered a multitude of pathways and mechanisms that contribute to the aging process at the molecular level. Recent advancements in molecular biology and genetics have provided unprecedented insights into the intricate workings of cellular aging, opening new avenues for intervention and potential rejuvenation. Among these, the concept of activating specific molecular targets to reverse aging has emerged as a promising frontier. Understanding the molecular basis of aging. Aging is a complex biological phenomenon influenced by a myriad of factors, including genetic predisposition, lifestyle choices, and environmental exposures. At its core, aging involves the gradual decline in cellular function and tissue integrity, leading to a heightened susceptibility to age-related diseases such as cancer, neurodegeneration, and cardiovascular disorders. Key molecular hallmarks of aging include genomic instability, telomere attrition, epigenetic alterations, loss of proteostasis, mitochondrial dysfunction, cellular senescence, stem cell exhaustion, and altered intercellular communication. These processes are interconnected and contribute synergistically to the overall aging phenotype. Researchers have demonstrated that therapeutically restoring youthful levels of a specific subunit of the telomerase enzyme can significantly reduce the signs and symptoms of aging in preclinical models. If these findings are confirmed in clinical studies, there may be therapeutic implications for age-related diseases such as Alzheimer's, Parkinson's, heart disease, and cancer. Researchers at the University of Texas MD Anderson Cancer Center have demonstrated that therapeutically restoring youthful levels of a specific subunit of the telomerase enzyme can significantly reduce the signs and symptoms of aging in preclinical models. If these findings are confirmed in clinical studies, there may be therapeutic implications for age-related diseases such as Alzheimer's, Parkinson's, heart disease, and cancer. The study, published today in Cell, identified a small molecule compound that restores physiological levels of telomerase reverse transcriptase, TERT, which normally is repressed with the onset of aging. Maintenance of TERT levels in aged lab models reduced cellular senescence and tissue inflammation, spurred new neuron formation with improved memory, and enhanced neuromuscular function, which increased strength and coordination. The researchers show that TERT functions not only to extend telomeres, but also acts as a transcription factor to affect the expression of many genes directing neurogenesis, learning and memory, cellular senescence, and inflammation. Epigenetic repression of TERT plays a major role in the cellular decline seen at the onset of aging by regulating genes involved in learning, memory, muscle performance, and inflammation, said corresponding author Ronald DePineux, MD, professor of cancer biology. By pharmacologically restoring youthful TERT levels, we reprogrammed expression of those genes, resulting in improved cognition and muscle performance while eliminating hallmarks linked to many age-related diseases. Aging is associated with various epigenetic changes that influence functional and physiological decline. One of the hallmarks of aging is the gradual shortening of telomeres, the chromosomal end structures that help maintain their stability. Free radicals also can modify and harm telomere sequences. When telomeres become extremely short or modified, they trigger a continual DNA damage response, which can lead to cell senescence, a phenomenon in which cells release inflammatory factors that can cause tissue damage, prompting aging and cancer. Telomerase is a protein complex responsible for synthesizing and extending telomeres. However, its activity is reduced over time due to the epigenetic silencing of TERT, particularly at the onset of natural aging or Alzheimer's and other age-related diseases. The DePineux laboratory previously showed that deactivating the TERT gene in vivo led to premature aging, which could be reversed through TERT reactivation. The researchers also observed that certain cells, such as neurons and cardiac cells, were rejuvenated without undergoing the normal cell division required to synthesize telomeres. Their observations led them to hypothesize that TERT had other functions beyond synthesizing telomeres and that overall telomerase levels were important in the aging process. Based on these findings, the researchers, led by DePineux and first author Hong-Suk Shim, Ph.D., aimed to develop a drug to restore TERT levels. Additionally, TAC treatment in human cell lines increased telomere synthesis with reduced DNA damage signal at telomeres and extended the proliferative potential of these cells, demonstrating the activity of TAC in ex vivo human models. These preclinical results are encouraging, as TAC is easily absorbed by all tissues, including the central nervous system. Yet further studies are needed to properly assess its safety and activity in long-term treatment strategies, Dopinu said. However, our deeper understanding of the molecular mechanisms driving the aging process has uncovered viable drug targets, allowing us to explore opportunities to intercept the causes of a variety of major age-related chronic diseases. Small molecule restores TERT levels, reversing hallmarks of aging. A high-throughput screen of over 650,000 compounds identified a small molecule TERT activating compound, TAC, that epigenetically derepresses the TERT gene and restores physiological expression present in young cells. In preclinical models equivalent to adults over age 75, TAC treatment for 6 months led to new neuron formation in the hippocampus, memory center, and improved performance in cognitive tests. Additionally, there was an increase in genes involved in learning, memory, and synaptic biology, consistent with TERT's ability to interact with and control the activity of transcription factor complexes regulating diverse genes. Recent research has identified several promising molecular targets and pathways that, when activated or modulated, have shown potential in reversing aspects of cellular aging. Sirtuins, these are a family of NAD+ dependent enzymes involved in regulating cellular metabolism and stress response. Activation of sirtuins has been linked to increased lifespan and improved health span in various model organisms. Compounds like resveratrol, found in red wine, have been studied for their sirtuin-activating properties. AMP-activated protein kinase, AMPK is a cellular energy sensor that regulates metabolic pathways in response to energy depletion. Activation of AMPK has been shown to enhance mitochondrial function, promote autophagy, cellular recycling, and improve overall cellular health. Targeting senescent cells, cellular senescence is a state of irreversible cell cycle arrest that contributes to tissue aging and age-related diseases. Strategies to selectively eliminate or modulate senescent cells, such as through senolytic drugs, have shown promise in rejuvenating tissues and extending lifespan in animal models. Mitochondrial function. Mitochondria are essential organelles involved in energy production and cellular metabolism. Declining mitochondrial function is a hallmark of aging. Interventions aimed at improving mitochondrial health, such as through mitochondrial antioxidants or mitophagy inducers, have been explored for their potential anti-aging effects. Epigenetic modifications, changes in gene expression patterns due to epigenetic alterations, play a crucial role in aging. Therapies targeting epigenetic regulators, such as DNA methyltransferases or histone deacetylases, hold promise for reversing age-related changes in gene expression and restoring youthful cellular function. Challenges and future directions. While the prospects of targeting molecular pathways to reverse aging are promising, several challenges remain. One of the primary hurdles is translating findings from preclinical studies in model organisms to safe and effective interventions in humans. Additionally, the complexity and multifaceted nature of aging necessitate a holistic approach that addresses the interconnectedness of different aging pathways. Future research efforts are likely to focus on identifying biomarkers of aging that can predict individual susceptibility to age-related diseases and response to anti-aging interventions. Advances in technologies such as CRISPR gene editing, single-cell sequencing, and artificial intelligence are expected to accelerate the discovery of novel molecular targets and therapeutic strategies. Conclusion. In conclusion, the pursuit of reversing aging through the activation of molecular targets represents a transformative area of biomedical research. By unraveling the intricate mechanisms underlying cellular aging and identifying key molecular players, researchers are paving the way for potential interventions that could not only extend lifespan but also improve health span and quality of life in aging populations. While challenges remain, the collective efforts of scientists worldwide continue to drive progress towards a future where aging may be viewed not as an inevitable process, but as a modifiable biological phenomenon. As we stand at the forefront of this exciting field, the promise of unlocking the secrets to longevity and vitality through molecular interventions offers hope for a healthier and more vibrant aging population in the decades to come.